Hello, everyone, and welcome to today's presentation on engineering the next generation of cells for protein biology. I am Lisa from Nexlum Bioscience, where we have a team of experts in the science of cell counting and cell-based assays, and I will be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. In addition, if you have any trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I am excited to present today's speaker, Dr. Bjørn Voldborg, Director of CHO Cell Line Development at the Technical University of Denmark. For a complete biography of Dr. Voldborg, please visit the Biography tab at the top of your screen. So without any further ado, I would like to hand it over to Dr. Voldborg.
Thank you very much, and thank you for giving me the opportunity to present our work here. It's always nice to present work, scientific work, to a good audience, and even better if it's something we're proud of, which I actually am. I'm pretty proud of what we've been doing here. So I'll be talking about the engineering of the next generation of CHO cells for protein production, the work that's been going on at the Technical University of Denmark for the past 8 years. So just very briefly on who we are, I'm working at the Novo Nordisk Foundation Center for Biosustainability at the Technical University of Denmark. It's important for me to stress that we're not affiliated with Novo Nordisk. We're not a part of Novo Nordisk. We're funded by the Novo Nordisk Foundation, which is a foundation like the Bill Gates Foundation in the US or the Wellcome Trust in the UK. Foundation that makes a lot of its funds from owning a big chunk of both Novo Nordisk and Novozymes, but it's giving the money with no strings attached into the scientific community and to academic research. The Center for Biosustainability is a center that is focused on the generation of cell factories. So basically we see cells as small factory buildings that are engineerable, that can be engineered to produce better what it's already producing or producing completely new products. We're doing this both in microbial cell factories, mainly to produce small chemical compounds, and then also in CHO cells, where the goal is to become better at producing therapeutic proteins. We want to do this, so this is being done in both with basic research. Half the funding, more or less, is going into doing classic basic research with groups of postdocs and PhD students. And then also the other half will be going into translational research, which is running core facilities and with staff scientists and technicians, where the goal here is to actually take the knowledge from the basic research groups and use that to generate cell lines and tools that can be used into industry and go out into the real non-academic world. Another big scope for us is the development of talents to educate postdocs, PhDs, and also to attract scientists into the Copenhagen and southern or Medicon Valley region of Denmark and southern Sweden. So the CHO program is funded by the Novo Nordisk Foundation. It was approved in May 2012, and we got a funding of about $60 million for 8 years, which is a good funding, I'd say. It consists of 4 scientific sections and 1 core section. And we are addressing the major challenges in the field. So when we started this program, we went out and we talked to pharma companies to see what do you really want. We have this program we are starting up, what would you like us to focus on? And we went to pharma companies, but if you look at the kind of the headings here, quantity, quality, time, that's probably what you would get as an answer no matter which industry you talk to. They want a lot of very good quality product fast. But if we split it up into more of the recombinant world, you see that there is some more specific areas to address. You want high titers, which makes it cheaper to produce. That can be addressed through transgene expression, translation, transport folding, cell viability, and so on. You want it to be able to purify it relatively quick and efficiently, to avoid immunogenicity degradation, to avoid losses in purification and contaminations. You want to be able to control the post-translation modifications, and this is something that we've been doing a lot of, and I'll be addressing that quite a bit later on. And then you would also like to do this quick, so get a quick development time and a stable expression over time. And we're looking into that as well. So the green words here are the areas that we've been focusing on in the program. So the CHO cells, what are they? They are the preferred host for the production of biotherapeutic glycoproteins. The reason for that is that they have this more or less the same post-translation modifications as the human proteins. They have human-like glycans, not completely, and I'll get back to that later as well. They have a good capacity for folding it correctly and secreting. They have a low transmission of viruses, high protein yield. They're highly adaptable. They are actually extremely adaptable, and you can basically do almost anything to CHO cells, and they will adapt along the way. They have a long FDA history. They originally were taken out of China back in 48 or 49, the hamsters themselves. And there was some discussions during the Cold War that there was a worry that the CIA and others, it was mainly China, worrying that they would be using these as hosts for biological warfare, like parachuting in small hamsters with diseases. I don't think that ever happened. You never know. But today they're actually become the host of protein drugs that have been used to cure millions of people around the globe. And that is the focus that I'm looking at, and that is why we really want to look at these to make better production cell lines. So the CHO cells are secreting cell lines. It's kind of funny because there were some colleagues of mine made a transcriptomic study of all human organs to try and see, okay, all human organs, what are they actually producing? And if you can see here, this is a graph of secreted proteins from different organs. Ovaries are actually really, really bad at secreting proteins. Another organ that is almost just as bad is the kidney, which is kind of weird when you first think of it, because this is CHO cells and HEK cells, which you would expect, which basically are the main two mammalian cell lines to produce secreted proteins. But when you think of it, back in the days, the problem here was actually not as much as getting a lot of protein, as even more to actually be able to purify it from the media. And having cell lines that secrete low levels of host cell proteins by themselves actually makes the media cleaner and makes it easier to produce, purify from. So this is the classical way of doing this. So people would have their in-house cell line. They would make a stable polyclonal pool by transfecting in their genes of interest. They would do some rounds of amplification to see if they can amplify the genes. They would single clone the screen for the best high producers, and then they would go and screen for an optimal bioprocess. And this can be a very lengthy process, and they would be doing this again for the next gene of interest. What we want to go is actually to have cell lines that are specialized for the production of different proteins. We want to be able to control the glycosylation. We want to have targeted integration sites. We want to have a robust bioprocess, a facilitated downstream processing, and a faster upstream development. Doing all this, you should be, if you think of it, I'd like to say that I would like the CHO cells to become almost a coli cell, except for the growth rate. But if you try and produce proteins from a coli cell, you would not start screening clones. You would assume that they are all producing more or less the same. And that's also what we're aiming at. Especially using targeted gene insertion sites, we're actually able to make cells that basically all will produce the same high levels of protein. So you will not have to screen for large batches of clones to figure out and get the best producer. The CHO program is an all-inclusive program. So we both have the bioinformatics side looking at all the omics, analyzing large amounts of data. We also have a glycosylation group that are looking into understanding the glycosylation. I'll be coming back to that. Protein production, phenotype, so on and so forth. The scientific sections are spread out over 3 universities on 2 continents. Actually, 4 universities on 3 countries on 2 continents. We have a genome scale modeling group at the University of California in San Diego, headed by Nathan Lewis, who is the bioinformatic groups. So they have been generating in silico models of CHO and analyzing a lot of this big data that we have been sending over there. We have a CHO cell line engineering and design group here at DTU. They have been designing a lot of the tools needed to engineer CHO, and they're looking very much into the investigation of the CHO secretory machinery, ER stress responses, product quality, and so on. We had a group at the University of Copenhagen that has been looking into understanding the glycosylation machinery and CHO cells, and then we have used these knowledges to improve the glycosylation machinery and engineer the glycosylation machinery of CHO cells. And then we had a group at the University of the Technical High School in the Royal Technical University in Stockholm, Sweden, that has expressed and purified the human secretome, basically more than 2,000 proteins expressed and purified from CHO very systematically, which have given us a very large data set of which proteins can be expressed in CHOs and which cannot. And then there's the CHO core, which is the translational part, which is headed by me. And we've been focusing a lot on making these cell lines. So we have been looking very much into making engineer the cell lines. We started out with a GMP-ready cell line and have been monitoring very tightly anything these cells have seen. So they should be ready relatively quickly to actually go back into GMP facilities if needed. We've been looking into glycosylation to increase the expression levels, to get a clean and virus-resistant cell line, and also try and eliminate process variants, especially lactate, that I will come back to. We've been doing this by running a high-throughput CHO cell line engineering platform, where we designed knock-in, knock-outs, and did the manipulations in high throughput. I'll get back to that in a minute. And then we analyze the cell lines using different approaches. We have expressed and purified a number of model proteins, and we're actually also producing therapeutic candidates by generating new drugs with specific tailor-made glycosylations on them. The knockout pipeline, this high-throughput pipeline of the CHO core that I've just mentioned for cell line engineering, especially gene knockouts, is looking like this. So we have the target identification, reagent design, which in this case is guide RNAs, as we're using CRISPRs. We use these we design the guide RNAs, clone them into high throughput, screen for the best guide RNA. We would typically clone more than 1 or 2, maybe 4 guide RNAs per target to pick the best ones. Then we prep them up, transfect into the CHO cells using a Cas9 that is fused to GFP. Then we can use the FACS to sort these out into 384 well plates. They grow there, they go onto a Celigo, and then to figure out which wells have actually growth in them. And then a robot picks them from the 384 well plates into 96 well plates. We grow them up, sacrifice 2/3 of them, 3/3 of them, 3/4 of them for next-gen sequencing to detect which ones have indels in the genes we want. And the rest, then with the last 25% will then be expanded, selected, and picked for either a rescue plate that goes into the freezer, or it gets expanded into shake flasks and banked. That way we have generated more than 700 different specifically engineered CHO cell lines over the years now. The cell lines are then getting analyzed, so we transfect them with model proteins, we select clones, we look at protein expression, we purify the protein, we characterize the proteins, we generate data for a number of omics analysis, and we also look at the phenotypes and growth, viability, and so on and so forth with the CHO cells. So when we want to use the cell lines as a factory building, as I mentioned before, we want to be able to control the protein production in the CHO cells. And to do this, we need to know which parts are in the factory. So we need to know which genome sequences are actually there. We published the genomes of the CHO and actually also of the original hamster some years ago. So we know what we have. We need to understand how the machines are talking together, how the genes, the proteins are talking together. So we've been doing a lot of modeling, or Nate has been doing a lot of modeling with his people in San Diego, to understand how the machinery and the network inside the cell line is actually looking. We have tried to understand the post-translational modifications, especially glycosylations through the Golgi, understand the metabolic networks within the CHO cells, so which metabolite goes where. And then we have spent quite a bit of time also to actually get the tools ready to be able to engineer all this. So with this, we've generated a number of different cell lines, and I'll go through some of them now. One of the first ones we did was actually to try and clean up the CHO. So we basically have been looking into which host cell proteins are actually swimming around when a CHO cell grows. And then we started knocking them out. So we took, first we knocked 6 out, then we added 5 on top of those, and at the end we added 3 more to clean up the CHO cells. And we can show that we actually, by doing just these 14, or actually maybe even just the 11, we actually reduced the whole cell protein load of the cells by about 50%. And we increased the viable cell density and actually also the viability over time by knocking out these 6, 11, and 14 genes. We've also looked into one of the things that have been a problem for especially bioprocess over a long time, which is the tendency for CHO cells to produce lactate when they grow. The lactate gets secreted out of the CHO cell, it lowers the pH, and then you typically add base to keep the pH constant. At some point, you change the osmolarity of the media, and that actually decreases the viability of the CHO cells in there. There's been a lot of work going on trying to remove these pathways to see if we could get rid of the lactate, especially by targeting the LDHA gene. But it has not really been done in a way that it wasn't possible to do them all, to knock out LDHA. It seems to be lethal. But we actually managed, by knocking out both LDHA and PDK1 to 4, we managed to knock out the LDHA gene, thereby generating a CHO cell line that does not produce any lactate, a CHO-Zilla. And the postdoc who designed this obviously went online and actually found a hamster dressed up as Godzilla, which we kind of took to our hearts. So this is the CHO-Zilla here. As you can see, it does not produce any lactate at all. If you look at the figure on the top right, the red line at the bottom is actually the lactate. So there is 0.0 lactate in this. It just doesn't produce any lactate. It shows higher viability and also reaches a higher viable cell density over time. So this is one of the other ones that we're actually pretty proud of. Then I'll be focusing the remainder of my talk mainly on the glycosylation. So we've been doing a lot of work on glycosylation machinery. Glycosylation of the therapeutic protein is important. It's a quality attribute that needs to be reported to the FDA. So when you get a drug that you put on the market, you have to deliver in a glyco profile, and you have to keep all batches you produce along the line, a long time. You need to keep it within these specs. glycans often a significant impact on the efficiency, activity, immunogenicity, stability, and the half-life of your drugs. So it matters. The group at the University of Copenhagen spent the first 4 years of the program to develop and understand and investigate understand the machinery of the CHO cells. And we use this knowledge to then engineer a set of CHO cells that actually can make tailor-made glycans. And we validated these on whole cell proteins and model proteins to show that we can actually make cell lines with tailor-made glycosylation. And this is just one example, the first one. So here we have knocked out 6 glycogenes. And if you look on the left side, you can see a glyco profile from wild-type CHO cells. And on the right side, we have knocked out 6 of the glycogenes that the CHO cell has. It can now only make biantenary glycans. We're analyzing this on the natural host cell proteins, secreted host cell proteins from the CHO cell. And we do that because we want to understand the glycosylation machinery here. We do not want to have influence on the of the protein itself. There might be biases towards which glycans is there room for in different, like antibodies would almost all of them only have biantenary because there's no room for anything larger. So this is on a variety of proteins, and that gives us a very robust idea of what the cell actually can make. And we've used this to make a full panel of cell lines. So we've made a large panel of cell lines that we can pick. You want a monoantenary, biantenary, triantenary, or tetraantenary, or you can have it without fucose, without sialic acid, galactosis, and so on. So it's trimmed both ways, which gives you a pretty good tool if you want to investigate glycosylation of your protein. And why would you want to do that? So the way you normally make a protein that you will go into clinical trials, you make a high producer CHO clone, and then you pick the best, often the best producer you have, which may give a very heterogeneous glycosylation site. So you have different versions of the protein with different glycans on it. But what you would actually really like is actually to be able to figure out which glycoform of these is actually the best. So if you take the panel that I just described before and express a little bit of your protein in each of these, you end up with up to 30 different glycovariants of your protein. You can then test that in your assays to pick the best glycoform you can that you want to go forward with, and then you take that and go into clinic. That same cell line can then actually be used to produce the proteins going forward and scaled up and actually end up going all the way to manufacturing with a cell line that will make the glycan you want and not the heterogeneous variants that you have. We've done this, so this is just a very rough example of what we've done. So this is 8 examples of how glycoprofiles look when we have used EPO into the cell lines. And in a shake flask, so there's nothing fancy here. There's no large bioprocess. And you can see that this is different variants of EPO. This is EPO with different glycans on it. And you can see that it's actually, you can pick and get your glycans with different, or get your protein with very defined glycostructures or glycoprofiles on them. We've done this with a number of proteins. One of the things that we figured out is there's a lot of, the liver is secreting quite a bit of protein into the plasma. And it turns out that most of the proteins produced by the liver actually have a very defined biantenary non-fucosylated glycoprofile. And this may have, or actually has, a very large effect on at least some of the proteins, maybe more than you'd think. So you need to have the real glycan, this biantenary glycoform on your protein to actually make it work. One example of this is Alpha1 Antitrypsin. Alpha1 Antitrypsin is a protein that protects the lungs from degradation from neutrophil elastase. It's a protease inhibitor. There's a number of patients who are born without Alpha-1 antitrypsin. Either they miss the gene or they have a variant that makes it never makes it out of the liver. And the symptoms they get is kind of like smoker's lungs. So you get this lung degraded over time. The treatment is augmentation therapy. So they go into the hospital typically once a week, get doses from 4 grams and up. I heard up to 12 grams of protein that they will need to get alpha-1 antitrypsin in. The products on the market are all derived from fractionated human plasma, and there is no recombinant proteins there. And why is that? So the AAT from CHO is, we looked at it, and if you look at alpha-1 antitrypsin or AAT expressed in normal CHO cells, you see a very heterogeneous profile. If you look at what gets out of the human plasma, it's, like I mentioned before, from the liver, very specific biantenary. And we had a cell line, as I showed before, that was actually pretty close to this. So we had a cell line that, in this case, was biantenary, still had the fucose on, and it didn't have sialic acids. And to make a long story short, we managed to make a cell line that can actually produce alpha-1 antitrypsin in the bottom right corner. This is recombinantly produced alpha-1 antitrypsin from our CHO cell, which is very similar to both the commercial drug on the market, which is derived from plasma, also very similar to plasma-derived AAT that we derived ourselves, or fractionated ourselves, and very different from when you produce AAT in wild-type CHO cells. We managed to pick some high producer clones of this and ended up into 1.4 grams per liter in the first media screen in a bioreactor. And this is stable over time. So after 63 days, we still have a cell line that can produce high levels of alpha-1 antitrypsin with the correct glyco profile on it. And we scale this up also to 5 milliliters, and we've shown that it actually, on an STS page and isoelectric focusing, it looks very, very much like the plasma-derived versions and very different from the CHO, wild-type CHO-derived versions. And there are some advantages of this. One of the things that you can start doing if you have a recombinant alpha-1 antitrypsin instead of plasma is you can start manipulating it. So you can either get, as I mentioned before, alternative glycosylations, which could be interesting for some diseases. You can add more glycosylation sites for improved half-life. You can do stabilizing mutation. You can actually even change the affinity. So if you do some mutations in the active site of alpha-1 antitrypsin, it suddenly gets very active again. It gets a very high affinity against furin instead of neutrophil elastase. So that might actually be a potential drug for other places where you would like to get rid of furin. Maybe like the very up-in-time COVID-19, where actually the spike protein needs a furin cleavage to be able to bind to the ACE receptor to infect cells. We can make a very low batch-to-batch variation, and the fact that you make it recombinant also means that we're not dependent on getting protein from patients who donate blood. You can formulate it, you can up-concentrate it, you can even add fusions to it. So you might even add targeting proteins or something to get it to go to the right organs or the right places in the body in the future, if that's what you want. So one experiment that we've done is we actually produce quite a bit of proteins. Here we have I mentioned 10 proteins that we have made and purified in all 30 cell lines, just to make different variants of these. And we're actually looking for more proteins. We will be starting up a protein production facility using, among other things, these glyco-engineered cell lines. So if anybody out there has the need for proteins with specific glycans on them that they would like to test out, please approach me on my email after this, and I'll be happy to talk further about how we can do this. So the future perspectives of this is that we are looking at completely new protein-based drugs. We have unnatural proteins. You can make fusions. You can make all kinds of different things that you could not do before. We can make CHO proteins that cannot be made in CHO today. We are looking into, we're making proteins with designed glycoprofiles, as I mentioned. And also by using targeted integration and other approaches, we're able to go much faster from bench to pharmacy, looking into cheaper development and cheaper production of therapeutic proteins going forward. With this, I'd like to acknowledge all the people involved in this. My group at the Technical University of Denmark, and also the program partners in the partners in the CHO program, Bernard Palton, who is the professor at UCSD, and he's also the grant holding PI, Jinmin Lee and Lise Grau from KAIST and DTU, Nathan Lewis from UCSD, Matthias Sylén and Johan Rockberg in Stockholm, and Henrik Clausen and Zhang Yang at the University of Copenhagen. And then we've had some collaborators, especially on the Alpha1 Antitrypsin, Richard Leduc at the University of Cherbourg in Canada, and Jerry McElvaney at the Beaumont Hospital in Ireland. And with that, I just want to acknowledge the funding and the support from the Novo Nordisk Foundation and DTU. And that's basically it. I'll be ready for questions in the Q&A session later.
Thanks, Dr. Voldborg. That was great, very informative. We already have a lot of great questions coming into the chat box for you. For now, I would like to introduce our second speaker, Dr. Henry Qazi, who is an application scientist here at Nexlam. For a complete biography on Dr. Qazi, please visit the Biography tab at the top of your screen. Dr. Qazi, you may now begin your presentation.
Thank you very much, Lisa. So this is going to be the instrumentation side of the discussion, specifically about the Celigo image cytometer and how it is used in the cell line development community for engineering the next generation of cells and protein production. Just as a brief background, for anyone who doesn't know who Nexlam Bioscience is, our motto is essentially that we want to deliver better tools for better biology. We have done this by, over the last 17 years, essentially designing and manufacturing cell-based analysis systems out of Boston, north of Boston, Massachusetts, and providing these image cytometry technologies. Our approach, along with just being a worldwide company, with myself located in San Diego, is essentially to have scientists helping scientists to increase accuracy and really to help them spread best practices with other labs. What this does essentially is bring scientists like myself. I have a background that includes doing CRISPR editing, along with doing single cell cloning, and pretty much doing that same type of work that Bjørn was discussing earlier. And it brings groups like me and him and other labs together where we can essentially collaborate. And we have several publications that are peer-reviewed in the cell line development community. So I welcome anyone to just explore the Nexlam website and to just further look at some of these publications. So the Celigo has been really integrated heavily into the cell line development community. We have been from the very beginning steps where you can start out with doing transfection, determining transfection efficiency of your cells, looking at the stability of those cells in terms of doing stabilized outgrowth, monitoring cells within wells, and identifying which populations to essentially move forward along with optimization of the whole process. Has been used by Celigo in the earlier stage, and then classically, the Celigo image cytometer is used to verify monoclonality, whether it's through limited dilutions or single cell dispensing, and then using it towards colony outgrowth. So just what is a Celigo? So the Celigo image cytometer essentially provides fast, time-saving, high-throughput analysis for imaging, and provides the ability to do like document monoclonality and expansion of your products with great ease. The goal is essentially to have an ability to essentially automate things, be able to scale up productivity without sacrificing validation of your data. And the Celigo allows for plate-based imaging with probably the fastest image cytometer in the market, which allows for accurate cell-based assays using both whole well bright field and fluorescence imaging, and it's mainly used as a screening tool within the cell line development community. The way the Celigo does this, and what's proprietary about the mechanics, is that it has what we call F-theta lenses and galvanometric mirrors, which essentially allow for speed to capture larger areas that you want imaged, and then as you look in the middle, accuracy going all the way to the edge of the well. We often say that Celigo picks up every cell in every well. Which allows you, whether you have adherent cells, suspension cells, whether you're doing colony outgrowth, and oftentimes when you're doing single cell dispensing or doing clonality, cells have this nice tendency to roll all the way to the edge of a well. The Celigo allows you to quickly and accurately pick up that single cell at the edge as well. The workflow is actually pretty simple when you think about the Celigo and how it can be used within the single cell sorting and single cell to colony growth. Essentially, you either use limited dilutions or you sort your cells into a plate of your choice, like a 96 well plate or a 384 well plate, put it into the Celigo, and the Celigo will do a whole well image, and it spits out your data for you. And then you can get these really nice heat maps, which I'll discuss in a second. The Celigo was essentially designed with the user in mind, so it's really designed for like a simplified workflow where you just go from you start, you put your plate in, you put your scan parameters on, you analyze the cell, do any kind of gating if you need to in terms of like doing population identification, if you're doing transfection efficiency, and then you get your results. And then the software can be simplified as simple as 2 or 3 clicks to get from introducing your plate to getting your results. And this is why the Celigo is very loved in the cell line development community. So the heat maps that I was discussing and wanted to share with you is, so when you get to the results stage and you want quick verification of what you have actually seeded and plated, when you're doing single cell seeding, a heat map can be generated, essentially creating a legend like on the right side, where you can say some things like, if I have more than 1 cell, mark it red. If I don't have a cell in there, mark it as yellow. This allows for color coding, visual confirmation of what you plated, and really aids in the entire workflow and helps with the accuracy of seeding. Panel C is an example where somebody essentially used the Celigo and fed back in towards optimizing their cell printing platform. So with limited dilutions, you can be very lucky if you get 1/3 of your population as single cells per well. And that's the example of that larger heat map that you see in the back. But what you can do is you can optimize your cell printer to essentially get towards an accuracy level of about 97%, according to this specific study. A few studies have shown that if you use both the combination of 2 instruments, both a cell sorter or printer and verify with the Celigo, you can essentially get an assurance of like 100% that you are actually successfully seeding a single cell and using a single cell to grow into a colony. Here's an example of using and getting a very quick information right from day 0. So one of the processes that is often recommended is essentially, instead of having to wait for 7 days, 10 days, or 2 weeks to see if I have a colony growing, is to quickly identify your single cells within that single well right away, so that you know what to expect and you can really develop your processes and be prepared to deal with those specific samples ahead of time. So this methodology, for example, uses actually a transient dye. And for transient dyes, it is often recommended that if you're doing any kind of fluorescent labeling, that it should be limited to reagents that do not interfere with the integrity of the cell line itself. So normally I recommend transient dyes such as CMFDA, which during single cell cloning, it doesn't interfere with the safety or overall product quality. And this has been shown through several studies. But if you can use fluorescence and bright field to analyze it, it really reinforces the assurance of monoclonality, especially for documentation purposes. So here's just a quick example where we had, this is showing one well out of our entire plate where they essentially did single cell sorting of both GFP and M. cherry cells, so mixed population, and tried to see and make sure that we did get successful analysis using fluorescent channel or bright field alone to verify single cells. And so here's an example where we see that indeed only one M-cherry cell was stained in that location where you can see on the left panel, and no other cells were present within this well. And visual verification is very easy to do within Cellego software. I did want to take this opportunity to mention that we did have a new application note that just came out called Modernize Your Cell Document Cell Development Assays with High Throughput Image Cytometry, and it is also an article that is available on Cell Culture Dish. Here's an image from that article, essentially showing the single cell plating within this well, for example. And plane A shows the use of a transient dye. And if you are not to use a transient dye, you can use GFP transfected cells or M-cherry cells, but usually those are recommended for non-clinical research. But if you need to be able to verify it in just bright field alone, panels B and C show the images in just bright field, a single cell, and its identification in panel C. And then you can essentially know right away that you had a successful seeding protocol instead of having to wait several days. And then a few days later, I believe this was in 4 or 5 days, in panel D, you see a nice colony growing in the bottom left of that well. That's an image overlay, like a color overlay over bright field, just for identification purposes, for just visual verification. And panel E and F show what that looks like, at least within the software, of identifying that single colony that arrived from a single cell. And so this process can be done in high throughput and is very easy to essentially identify those single cells to single colony growths. I would be remiss if I did not mention new developments within the cell line development community. Many researchers and many sites have actually started using our Celica high throughput cell counter in the middle, which essentially allows them to count 24 samples in 3 minutes or less. It's become really useful in cell line engineering, processes, production, manufacturing. It's been a relief for a lot of people that have had existing bottlenecks, where now they can count hundreds of samples within minutes instead of hours. And for people that do CHO cell work, oftentimes you can bring that down to taking 24 samples and counting them in about a minute. So it's actually become a very successful instrument within the cell line development community. And essentially, it's a dedicated cell counter, which really helps just move samples along quickly, checking for viability and cell counts. The other thing I did want to mention is just Nexcelom Bioscience's role overall. We are unique in providing support and resources to the cell line development community. Nexcelom Bioscience is committed to supporting the community by providing documentation, assays, protocols, and essentially is providing a complete image cytometry solution to advance research and therapeutics as best as we can. And at the very basic level, you have access to scientists like myself who have done this type of research, and we can even give recommendations of what type of plates to use and what is popular in the field for your specific application if you need it. So overall, the Cellego provides fast, time-saving, high-throughput analysis and imaging solutions with the ability to essentially document monoclonality and expansion with great ease. And you can use the Cellego not just for just single cell to colony outgrowth, but for lots of other applications which are available to be explored by yourself at nexcelom.com. With that, that is the end of my presentation. I will take any questions or hand it over to you, Lisa.
Thanks, Henry. That was great. We already have questions coming in. Everybody feel free to put in questions into the chat box. And for the first question, it's for you, Henry. Can these cell counters count primary cells?
Actually, they can. So what is really great with primary cells is you can use nuclear stain dyes. We recommend acridine orange and propionate iodide, which essentially gets rid of any of those background signals that you might have from primary cells. Debris, all of that gets ignored, and you get your very accurate live dead counts. And the Celica is essentially designed to do this in high throughput.
Awesome. Next question is for you, Dr. Voldborg. Is it possible, and thus are there efforts to humanize the glycosylation pattern in CHO cells?
Yes, it's not only possible, and there are efforts actually. So it's one of the things I realized that I skipped too quickly over. The Alpha1 Antitrypsin variants that we are making are actually made using a humanized glycoform, where it's typically is the question here is the sialic acids, whether it's a 2,3 or a 1,3 or a 2,6 sialic acids, and 2,6 are the ones that are prevalent in humans. And that is what we knocked out the 1,3 sialic acid transferases and put in the human one. So these are making humanized glycans. So the answer is yes.
Great. So the next question is also for you, Dr. Voldborg. Great talk, they say. I was wondering if the production of HIV-1 proteins, specifically the integrase protein, could be performed with these cells.
Probably. I don't know about the integrase protein a lot, so it's a bit hard to say. If it's a secreted protein naturally, then it would probably be glycosylated and then be interesting for to produce in CHO cells. But if it's an integrase, I guess it's working intracellularly, so it might not be. The question is, I don't know. I don't know not enough about the protein for this.
Okay. How accurate is the single cell GFP sort? Do you manually check your single cell and single well seeding by eye? That's for Dr. Voldborg.
Yeah, no, we don't. We use a Celigo. So we believe that the single cell sort is actually pretty accurate. The GFP is, as I mentioned, fused to the Cas9. So we mainly use it to enrich for the transfected cells. So if there's no Cas9 in the cell, it's not relevant, because then it's not going to get gene edited for us. And then they get single cell sorted out into 384 well plates, and then we use a Celigo to figure out which wells have growth in them. And then we pick from that. We don't look specifically whether it's a single cell or not. We would figure it out when we look at the number of genetic engineering, genetic edits. If we get too many different ones, it's probably more than one clone, and then we would discard it.
Okay. You mentioned that historically it was an issue that cell lines had too much secreted proteins in the medium. How do you select for high producer secretor cell clones of specifically your protein of interest, but not other proteins? Or is it that no longer an issue like it was historically?
Historically, it's not as much an issue as it was, because the purification techniques have improved dramatically since. So it's been a lot easier to produce purified protein out of this media. But we are looking into it. Normally, what we see is that we get about 0.5 milligram of secreted host cell proteins in the media. And you would normally, when you look for high producer clones, you would be looking at cells that are producing almost grams per liter or more. So in that case, it's negligible. You don't look for ones with very little host cell proteins. But by removing them, as we've done in one experiment on one cell line, it gets easier you get fewer co-purifying proteins down the line. So it's more like proteins that tend to stick to your protein, to your protein, or that gets problematic when you need to purify it away. Those are the ones that we removed.
Okay. Do you primarily use CHO cells for all the engineering, or do you use other cell lines such as SF9 or HEC293?
Well, the CHO program is a CHO program, so it's we mainly looked at CHO cells, but we've done actually some work in HEC as well. So we made actually a HEC-Zila version as well. But the focus has been on the CHO, and that's because the focus of the whole program is on CHO and the use of CHO cells for production of therapeutic protein. It's the main workhorse used by industry, so that's why we focused on that.
Okay, this next person says, thank you for these great presentations. A couple of questions. So the first one is, which purification methodology do you use, and which provides optimum yield for recombinant protein production?
Well, we use chromatography, so it's basically a column purification. The specific methodology of which columns depend obviously on the protein. If we can stick a tag on it, his tag for instance, then we can use specific nickel columns to purify using the his tag. If it's antibodies, there's specific protein A columns for that. If we, like for the Alpha1 Antitrypsin, where you cannot have tags on it, we developed a 3-step purification protocol using some ion exchange and size exclusion to get a reasonably pure protein.
Okay, the second part of the question was, how would you validate proteins' functional conformation and post-translational modification within your methods?
So the functional conformation, we try to see if we can make an activity assay. So with the Alpha1 antitrypsin, we test it to see whether it can inhibit elastase in an in vitro assay. It's actually a fairly simple one. If it's an antibody, we can look at the affinity that it still will bind to its target. And post-translation modifications, we're mainly looking at glycosylation. And the glycosylation we validate using fluorescently labeled, we shave off the glycans, label them fluorescently and run them on a mass spec. And then we get the profiles giving the figures that I showed in my presentation.
Okay, another question for Dr. Voldborg. Have there been any issues with cryopreservation after CHO lines have been produced? Do you see this being important down the line?
We have not seen any problems with cryopreservation. I mean, it's just we store them in liquid nitrogen like you would store any other CHO cells, and we so far have not seemed to meddle with anything that would make them hard to store there. That would be, you could imagine that you could end up doing that. Some of the glyco-engineered cell lines are actually hard to transfect if you use electroporation, but that's another story. That's probably because by altering sialic acid levels, you actually alter the charge of the cell, and thereby the standard protocols for electroporation of cells will not work anymore because it looks differently from an electric point of view.
Okay. In addition to VCD, pH, and temperature, what would be the top several variables of a CHO culture to actively monitor for a successful batch?
VCD. Get the highest VCD you can get. That's the one that then you get the highest production.
Okay, so this last person says, it is really interesting how we can use proteins as treatment. I have no experience on that, but it does look interesting. At the end of the day, the molecular biology dogma. Congrats, and I hope the good job keeps coming. I know this may be a little off topic, but do you have any knowledge on PVDF and its potential use for protein adhesion outside of Western blot?
The question here, the answer is no. I don't have any knowledge on what it's been used. I could imagine that you could use it if you want to get rid of old proteins from some media or whatever. But it's extremely, as you know, it's very unspecific. It will bind any protein at all. So you cannot use it to select for the protein you want.
Okay, I have one more question that came in. What is the theoretical limit of CHO for IgG production?
That's a good question. If you look at if you take it a step further and look at what a plasma cell produces in the human body, then we're talking 200 picograms per cell per day. What we can reach in a CHO cell is about a quarter of that, I would imagine, maybe even less. I think I've heard about 15 to 20 grams per liter in really optimized, where you really have spent a lot of time optimizing the bioprocesses and getting the highest producer and so on in an industrial setting. I mean, it can probably be expanded much more if we really put efforts into it.
Okay, It looks like that's all of our questions we have for today. Thank you both for great presentations, and thank you to our audience for taking the time to attend today's presentation. Questions that we were not able to answer during today's session, if you have any additional ones, we can answer them via email using the contact information you submitted when you registered for the event. We do ask that you complete a short survey at the close of the webinar. Your thoughts and comments are greatly appreciated. And also make sure you visit the poster hall and exhibit booths where you can learn more about a variety of exciting technologies. This webcast will be available to be viewed on demand as it has been recorded today. So until next time, we hope you have a great day and enjoy the Cell-Based Assays Virtual Symposium. Thank you.